Vaccines are often associated with scientific breakthroughs, but behind every vaccine is a quieter challenge: growing enough virus safely, reliably, and at scale. Without suitable cells, vaccine production can slow from weeks to months, increasing costs and limiting supply. This challenge is especially critical for animal and aquaculture vaccines, where disease outbreaks can affect food supply and regional economies. At Taiwan's National Pingtung University of Science and Technology, Professor Guan-Ming Ke has spent more than a decade working on this hidden problem. His motivation was clear: to improve vaccine development without relying on genetic modification or sacrificing animals. Instead of redesigning the factory, Professor Ke uses a different strategy: auditioning the workers and building the right workplace. Thousands of individual cells are screened to identify those that naturally perform best, much like selecting the strongest workers for the job.
然后我们也因此就是开发出用细胞，然后协助疫苗厂去开发出很多虽然全病毒的，然后非常低成本的一些，可是又有效的。
Those cells are then placed in carefully controlled environments, given the right nutrients, conditions, and monitoring to perform consistently over time. This non-genetically modified approach means the cell's DNA is not deliberately altered in the laboratory. Using this platform, ordinary cells become high-efficiency virus factories, producing millions to tens of millions of virus particles per production cycle. The method works across diseases affecting pigs, poultry, fish, and dengue virus, showing flexibility beyond a single pathogen. An additional benefit is ethical as well as practical. Growing viruses in stable cell lines reduces the need to propagate viruses in live animals. This helps advance vaccine research without sacrificing animals, while also improving consistency between batches. Cost and scalability are addressed too. These cells can grow without animal serum, which often accounts for 30 to 50% of cell culture costs. They can also be expanded in large, free-floating batches, suitable for bioreactors holding hundreds to thousands of liters. Together, this platform reduces cost, risk, and development time, helping vaccines move from lab to real-world production. Looking ahead, Professor Ke sees this platform as a foundation for faster responses to animal disease outbreaks and stronger biosecurity. At NPUST, this work reflects a broader mission: connecting research with real-world agricultural needs.